Hello everyone, my name is Sabrina Daglish and I'll be presenting a poster done by myself and Dr. Christopher Thompson called Acai and Aronia Inhibit Promyelocytic Differentiation into Neutrophils. So Acai and Aronia are both considered complementary and alternative medicines. These medicines can be used in conjunction with or as an alternative to conventional treatments. Complementary and alternative medicines, also called CAMs, are not thought of as conventional treatments because they haven't been as widely studied, and therefore we need to learn more about how these treatments affect the body and how they can benefit us. Acai, this berry here, and Aronia, this berry here, are both considered CAMs that are anti-inflammatory and immune boosting due to their high levels of antioxidants. So we sought to determine if these berries truly are immune boosting, and we focus on one specific aspect of the immune system called neutrophil differentiation. So neutrophils are the first responders to the site of infection, and therefore are very important for mounting an immune response. However, they only last in the blood for a couple of days before undergoing apoptosis, and because of this, they have to be replenished constantly in a process called neutrophil differentiation. So this differentiation process is important for being able to mount an effective immune response. Once you reach the end of the differentiation process, you get a fully functional neutrophil that can do everything that it needs to do. So we measured neutrophil differentiation by looking at when these specific functions of the neutrophil develop during the process. So the first thing that we can measure is called actin rosettes. So in this image here, actin is stained red, and we can see that it's very concentrated on the outside of the cell, and we call this a rosette. So these actin rosettes are an indication of the cell being able to move, so high motility, and actin polymerization. So this starts to show up during the myelocyte stage. And the other thing that we can look at starts to show up in the metamyelocyte stage, and it's called phagocytosis. So phagocytosis is when a neutrophil will wrap itself around a pathogen and engulf it in order to try to kill it. However, the neutrophil can't actually kill this pathogen until it becomes a banned neutrophil when it develops these oxygen-dependent killing abilities, which are associated with the ability to produce reactive oxygen species, which are the actual chemicals that will kill the pathogen that's been phagocytosed. So the final step of differentiation is the segmented neutrophil, and this is a fully functional neutrophil. So in order to measure differentiation, we use the HL60 cell line. These cells are stuck in the promyelocytic stage, but they can be induced to differentiate with two different chemicals called DMSO and DMF, and we call these differentiation agents. So in order to make our results as physiologically relevant as possible, we used a simulated digest fluid that mimics the way our body would digest acai and aronia. This simulated digest was added to our treatments of the HL60 cells during differentiation. So moving on to our results now. In figure 1, we compared undifferentiated HL60s to DMSO and DMF differentiated HL60 cells. So as you can see, there's a lot more red or actin staining in our DMSO and DMF cells as compared to our control. So this tells us that these actin rosettes that we're seeing are a good way of measuring differentiation. From these confocal images, you can also look at phagocytosis. So we have our actin staining in red, and we have bacteria that are producing GFP, so they will appear green on confocal. So when actin and bacteria are in the same place at the same time, that is when phagocytosis is occurring, and the confocal microscope will tag this co-localization as yellow. So yellow color indicates phagocytosed bacteria. We can see this pretty well in this DMF cell here. So the quantification of our results are in figure 2, and you can see we have high levels of both differentiation markers for DMSO and DMF treated cells. And this tells us that both actin rosettes and phagocytosis are valid ways of measuring differentiation. However, we noticed that about 16% of our control cells are still able to phagocytose. So we wanted to do a microbicidal assay to see if these control cells are actually killing the bacteria that they're phagocytosing. So in figure 3, we did a microbicidal assay where we exposed our HL60 cells to Staph aureus bacteria for an hour. After that, we lysed these cells so that we could culture the bacteria and see how many were killed. So we counted each colony, which represents one bacteria that survived, and we saw a lot less colonies in the DMSO and DMF treated cells, indicating that these HL60 cells are killing bacteria more than the control. So again, now we know that actin rosettes of phagocytosis are effective ways of measuring differentiation. So moving on to figure 4, we actually added our berries to see if they have an immune boosting effect now. So we have our control DMSO, and we have our positive control, which is DMSO and just our simulated digest fluid. Then we have DMSO in our digested acai and DMSO and digested aronia. So it's hard to see trends based on these confocal images. However, it's important to note that in the aronia treatment, we have some normal cells, such as this one with a nice actin rosette, but we do have some with hardly any, if all, actin staining, and this is an indication of cell death. So looking at the quantification of our results, we have a strange results with our DMSO, which, as you can see from figure 2, is not very similar with the level of differentiation markers. However, this is probably just because we had a hard time finding cells for this slide. We only counted 23 cells, so we clearly need to do more trials of this in order to get more relevant results. However, with our DMSO and simulated digest, we see numbers that we would expect with high levels of differentiation markers. With DMSO and acai, we see a decrease in these markers slightly. However, we need to do more trials to determine if this is significant. And then with DMSO and aronia, we see a pretty drastic decrease in the number of differentiation markers. So as you notice, or as I pointed out, we have these dead cells and we need to figure out why aronia is killing these cells. So we have 2 theories. The first theory is that aronia is simply cytotoxic to the cells, and we've actually seen that this is the case in raw macrophages. However, it's also possible that aronia is differentiating the cells so quickly that by the time we look at them after 5 days, the cells are already starting to undergo apoptosis. So we need to do more experiments in order to determine which of these is the case, what aronia is really doing to these cells. So in conclusion, our results suggest that acai and aronia might decrease neutrophil differentiation. What this means is that if this is the case, they certainly would be anti-inflammatory, because if there's less neutrophils in the blood, then there's less of an ability to mount an immune response. However, this certainly wouldn't be immune boosting if the berries decreased neutrophil differentiation, because if there's less neutrophils in the blood, then it's harder for the body to mount its immune response. So these companies are clearly a little misleading about how they market acai and aronia. If you would like to know more about CAMs, acai, aronia, or neutrophils, I have a written up introduction, and you can view it on this QR code, and you can also view my references there. So thank you all for listening.